Ashley. I'm studying chemistry, and this summer I've been making and researching copper catalysts. So I'm going to start with talking about photosynthesis. Photosynthesis is one of the most important biological or chemical reactions. It's the foundation for all life, so it's pretty important. Part of photosynthesis involves the formation of carbon-carbon bonds, and this hasn't been successfully replicated in a lab. So nobody has done the photosynthetic process artificially. So why is this important to study? It's important to understand these fundamental processes in science. There are a lot of exciting potential applications for artificial photosynthetic cells. And what I mean by that is a cell where you can take air that is not usable and recycle it into products that are usable. And this would be great in a closed system where it's not practical to have, say, a tree, like on a spaceship or in a submarine. And similar molecules to the ones that I was trying to make have been used to create superconducting cells, which is also really cool. So carbon-carbon bonds in nature. During photosynthesis, the products you get are oxygen and carbohydrates or sugars. So the net reaction is you have carbon dioxide plus water and energy, and in nature that's from the sun. And then you get your carbohydrates like glucose and oxygen. And part of these mechanisms involve carbon-carbon bonds forming. This is just the overall umbrella reaction, but there are lots of smaller reactions that go along in that. And before you form carbon-carbon bonds, you have to have an extra electron on that carbon dioxide. So a molecule with an extra electron on it is not happy. It's known as a radical, and these are like the free radicals in your body, like why you eat antioxidants, so that you don't have these free radicals going around your body and hurting your cells, because they're so reactive. So the same kind of idea, highly reactive, unpaired electron. And since these molecules are so reactive, if you have two of them, they should meet up and form a bond. So you have these dots represent electrons, and all the ones that are pairs, they're nice and happy, they're stable, they're not going to react most of the time. And the red dots are the unpaired electrons. So if you have two unpaired molecules with unpaired electrons or radicals, they will find each other and form a bond. So that's two electrons coming together, and they're happy and stable, and everybody likes that. So, to summarize that introduction, in nature, photosynthesis results in carbon-carbon bond formation. The first step for those carbon-carbon bonds to form is to give carbon dioxide an extra electron, or to make it a radical. And the ultimate goal of my research was to make compounds that will mimic this process artificially in the lab. So, synthesizing the compounds. Ligands are molecules that bind to metals. I was using copper as my metal, and the ligands I were using, or the molecules that were binding to my copper, were called 2,2'-dipyridylamine, or DPN, and di2-pyridyl ketone, or DPK. These are molecules that you buy already made. So my DPN looks like this. This is one of the ligands, or the molecules that binds to copper, and the other one was DPK, and it looks like that. And you can see that they're very similar looking, or similar in structure. So why was I using these ligands, or these molecules to bind to my copper? Previous research in our labs has made molecules with DPK using platinum instead of copper, and these have been reactive in the presence of CO2. So it made sense to use these molecules to make CO2 catalysts again. So why was I using copper instead of platinum if platinum had already worked? Well, there's a couple reasons. Copper is a lot less expensive than platinum, a lot less expensive. And it's also less toxic to people. So those are two pretty good reasons to try it with copper, since we already know it has some similar properties. And so why DPN if we'd already seen that DPK had success? Well, DPN is so similar in structure that it's reasonable to expect it to act similarly to DPK. Another reason to use copper is that it's been shown from the scientific literature to form clusters, which is not normal. It's a very weird thing for chemicals to do. But you can see there's copper there, a copper there, there's copper over there, and copper over there, and this copper over here with 2 ligands on it. This picture doesn't show the DPK ligands attached to the copper, but you can see it's kind of a big cluster, and so that's really interesting and unusual. So since in clusters all the atoms are really close to each other, if we bind carbon dioxide to one of these clusters, then when they leave as radicals, hopefully, they'll be really close to each other. And again, if you have 2 carbon dioxide radicals close to each other, they should meet up and form a bond. And then you have carbon-carbon bonds. So when I started this summer, I had a lot of choices. I had copper +1 and copper +2, which are really similar, but a little bit different in how their electrons are arranged. And I realized pretty quickly that I wasn't going to have time in 8 weeks to study all of the compounds I could make with these. So I thought that copper +1 would make a better electrocatalyst, because when it's given an electron, it ends up unpaired, so it's kind of like a radical. And if it's kind of like a radical, it's more likely to give that electron away so that it can become more stable. And hopefully it will give that electron away to carbon dioxide, so carbon dioxide becomes a radical, which is what we want. So these were the final 7 compounds that I ended up with. I chose these because I could consistently reproduce them, and they had really good solubility properties, which means it was easy to deal with them and get them to dissolve in things. So I had a ratio of copper DPK 1:1, copper DPK 1:2, and copper DPN 1:2. And this picture is a mixture of these 2 products right there. So they're all really pretty colors. And here are samples of my products. So we're going to move on to catalysts. Electrocatalysts are substances that increase the rate of a chemical reaction without being consumed by it. Getting an extra electron on a molecule or making it into a radical requires a push, because like I said, radicals aren't stable, so they're not just going to form all on their own. In nature, sunlight provides this push. You get energy from the sun, and that allows these radicals to form. But I was using electrical energy in the lab. CO2 catalysis testing. Once I had made all the products, I could start running tests on them to see if they were good carbon dioxide catalysts. My main tool was cyclic voltammetry, or a CV scan, which measures the electron movement, also known as the current, through a solution while varying the voltage to an electrode. So I'm going to go over the catalytic cycle. This is a graphic of what I hope to happen when my compound is in the presence of CO2. So I have the copper DPK molecule that I make, and it's in the presence of CO2, so it's bound to CO2. I'm running electricity through a solution of this. So that's my electron. That comes from the electricity. So that electron is going to move onto my copper. And then that electron is going to move onto the CO2, and then the CO2 is going to leave as a radical. That leaves the copper free to react with CO2 again, go back to the original place, and accept another electron. So this will get kind of like a domino effect, and this is how it's acting as a catalyst because it will greatly increase the current. So I'm going to go through that same thing minus the graphic. The catalyst, or my copper DPK molecule, will bind to CO2. Then the catalyst will take an electron from the electrode, put it on the CO2, then my carbon dioxide, or CO2, will leave, and that leaves my copper free to react with carbon dioxide again, and the whole cycle can repeat over. So it's kind of like a domino effect. So a carbon dioxide catalyst will generate more current when carbon dioxide is present. So if I have my copper DPK compound in a solution, so this would be a solution containing one of my compounds, and these are electrodes, and I'm running electricity through this. This is a close-up of an electrode. It's just a graphic, but so if there's no carbon dioxide present, this first scenario is what I would expect to see happen. An electron goes from the electrode onto my copper and DPK, and then it's done. That copper is done taking electrodes, so that'll produce some current, but not very much, because you just have the electron to the copper, and then another copper can come, an electron to the copper, and like I said, that produces a small amount of current. However, if my compound is in the presence of CO2, what I should expect to happen is an electron move from the electrode to the copper to the CO2, and then the carbon dioxide can leave as a radical, like I showed you in the previous graphic. So if I'm making a bunch of these carbon dioxide radicals, they can hook up, and I have generated current and hopefully made carbon-carbon bonds. So now I'm going to move on to some of my actual data. This is data I got from running a CV with the copper DPK 1 to 2. On this axis, we have potential, which is measuring the voltage, which is the same kind of voltage you have in a 9-volt battery, and that is determined by the software. And what I'm actually measuring is the current, which is on this axis. This is one of my compounds in solution without the presence of CO2. So this is what I would expect to happen from the previous graphic. The electron moving from the electrode to the copper and it being done. So the current increases here a little bit if we start at 0, and then that's just electrons moving onto the electrode. And then this jump in current is the electrons moving from the electrode to the copper. So you get some current, but it's nothing huge. And this peak right here is just water, so you can ignore that. And then I reverse the scan, and these bumps here are electrons moving from the copper back onto the electrode. So when I add CO2, I get a different result. The red line is carbon dioxide. So if I start at 0, almost immediately you can see the current rise. And then it goes up again, and it goes up again. So this is kind of that chain reaction or domino effect that I was describing earlier, where the electron is moving from the electrode to the copper, to the CO2, and then the CO2 is leaving as a radical. So this is really exciting. This is exactly what I want to see. Not only is the height of the red peak here indicative of how much current was flowing, but also the entire area under this red line is how many electrons are moving. So if you compare all of that area under the red line to all of the area under the blue line, that's a pretty big difference. The other thing is, and it's subtle, but when I reverse the scan with the carbon dioxide, these bumps go away that we saw in the first one. That means that the electron can't go from the copper back onto the electrode because it's gone. It's no longer on the copper, which means that that electron left with the CO2. That means I'm making CO2 radicals in my solution, which is exactly what I want, because if I'm making CO2 radicals, I could potentially be making carbon-carbon bonds. So that's really exciting. So I saw some success with my DPK. The 1 to 2 that I just showed you had the best results, but I had other compounds that also showed good results. There was a clear increase when my compound was in the presence of carbon dioxide, and we know that it was reacting with carbon dioxide because when I added nitrogen, or the inert gas I was using, back to the system and got rid of the CO2, the results went back to what they were in the first place. So we know it was specifically reacting with carbon dioxide. Here's a picture of that compound that I just showed you the data of. Again, they're really pretty. And to analyze this a little bit more, on the same catalyst, I would get a lot of bubbles, especially if I ran the scan slowly. And although bubbles are really annoying when you're running CV scans because they can disrupt your electrodes, it's really exciting because I'm making a gas. So is that oxygen or carbon monoxide? We're not sure because we don't have the equipment to actually gather the gas and test it, but it's still something that's definitely worth further investigation. Another main part of my research this summer was characterizing what I had made, because when you mix chemicals together, it's not exactly 1 + 2 = 3. You sometimes, or usually, don't know exactly what you're making. So I did standard characterization tests like IR and NMR, and that's like, an IR is infrared spectroscopy, which is like a fingerprint for a molecule, and NMR is nuclear magnetic resonance spectroscopy, which is like an MRI for molecules. So it just tells me more about the structure and what I have. What I really want to do is obtain X-ray crystal structures, because those are 3D snapshots of the molecule I have, which means they show me what atoms I have, where they are, how long the bonds are, and just the whole, what it looks like, how everything is bonding, which is really valuable information, because it's helpful in understanding how the metal and the ligand are interacting. And if we understand more about that, we can tweak the synthesis to hopefully make a more effective catalyst that creates more current in the presence of CO2. I do have a partial crystal structure of one of my compounds. It's one of the 1-to-1 ratio with copper and DPK. These are 4 copper molecules, and it actually is a cluster, like we saw in some of the literature earlier. And clusters are good because you have all of these atoms very close to each other. The carbon dioxide is probably bound in there somewhere. And if you have 2 carbon dioxide radicals really close to each other and they meet up, they'll meet up very easily because they're right next to each other. These areas represent my ligand bound to the metal, or the DPK. And these in here are actually, these 2 are carbon monoxides. And this is really interesting because we made this under pressure from carbon dioxide, and this means it went from carbon dioxide to carbon monoxide. So there's an oxygen missing somewhere. So where did that oxygen go? Maybe it went into the air. That'd be really interesting. So again, worth further investigation. And this is a picture of one of my DPN crystals that I grew. They're really cool. And there's a couple products in there, and it's really a pain to separate them because you have to look under a microscope and use tweezers, so kind of tedious. And this is a picture of the molecule I just showed you the partial crystal structure of. So this is what it looks like in real life. So to summarize, I made compounds that were actually CO2 catalysts that produce more current in the presence of carbon dioxide, which means that I was hopefully making carbon dioxide radicals that were forming carbon-carbon bonds, which is exactly what we were looking for, because that's what happens in photosynthesis. But there's still a lot of work to be done. Further work and research includes, for me, publishing a paper with the crystal structure I have. I'm expecting more crystal structures because we delivered some to UVA, and we're just waiting for them. So obtaining the rest of the crystal structures and growing some of these crystals under carbon dioxide. And that's important because if we grow them under carbon dioxide and they have carbon dioxide in the crystal structure, we can see exactly how it's binding and what interactions are going on there. It would be interesting to experiment with other metal centers, like nickel, because they're similar, kind of like the platinum is similar, but they could have some different properties. And the copper-2 chloride compounds that I didn't have time to look at this summer had good preliminary CV results, but again, I just didn't have time to do all of it. So, I'd like to thank Julie Hemstreet for organizing everything, the honors committee, and Dr. Rob for being the best faculty sponsor ever, and teaching me how to use all the equipment because I didn't know how to do anything, and the Sweet Briar Chemistry Department for letting me use all their equipment and chemicals, and their labs. So, are there any questions?
question?
Couldn't you tell if you made oxalate then? If you would think it correct?
Some of the platinum compounds that have been made here before actually did make oxalate. I made solids a couple of times, and I was only able to run an IR on one of them, and it was not oxalate. But I didn't have time to. I would have had to set up like a bulk electrolysis cell and collect more of the product, and I just didn't have time to do that.
So you were making though the free radicals that you thought if they were close together would form oxalate. So.
And there's other simple or carbon bonded molecules that they could be making as well. I had solids form on my electrodes a couple of times, but another problem I had was I would take my solution that had solid in it and dump it onto a watch glass, but of course it would still have the compound that I was testing in it along with the extra solid. So I, again, didn't have time to filter it and all of those things. Yeah.
Can you go back to the partial crystal structure that you have?
Sorry.
Okay. So, and this is your 1 to 2?
This is 1 to 1.
The 1 to 1, okay. But you said the 1 to 2 work the best. Is that right?
The 1 to 2 is the best catalyst.
Okay. Why do you think the 1 to 2 work the best out of those crazy ideas?
I'm not sure. I wish I had a crystal structure of it, because that would tell us a lot about what's going on, but I don't have that yet.
Yeah, looking at this structure, clearly, well, when you synthesize the compound, so you've got the carbon.
I don't think that that other product, the 1 to 2, I don't think it's a cluster. So maybe if it's mononuclear, that might have something to do with it. Maybe the cluster thing is, maybe that's not true. Although this worked pretty well too, so it's hard to say without having the structure.
So if it's not in a cluster, then that might make it a better catalyst. But then would that also perhaps make it worse at actually combining to make the carbon bond because they wouldn't have the proximity issue, which is further apart? Is it one of those things where it kind of goes both directions? Because you wanted the cluster for it to be close together. So I assume if you think it's not a cluster, you think that they're farther apart. Is that?
If it's not a cluster, then yeah. Supposedly, the carbon dioxide would come off of one molecule, and it would come off another molecule over here, and it would have a harder time finding and meeting up. Does that answer?
Yeah, it feels like a catch-22 kind of.
Yeah. So, one of the problems I had was these clusters are really hard to get into solution. So I wasn't able. The number 1 product, the 1-to-1 copper GPK, we're almost positive that's a cluster because it's so hard to solvate. But because it's so hard to solvate, I never got it into solution to run a CD on. So it was that difficulty caused it hard to do like a juxtaposition of whether or not clusters were working better than the mononuclear ones. And again, I don't have the crystal structure, so maybe the other one's not even mononuclear, or yeah, just has 1 millicenter.
Do you think that copper has more potential than a platinum at this point? Is there any way to know that in the long term? Or?
I don't know I don't know what kind of current the platinum compounds generated. Maybe. Do you know?
Yeah. We see a lot of gas evolution for copper compounds, and we didn't see it with platinums. And so that's encouraging. But we did make a polymer, a linear polymer with the platinum that when we tried some of these experiments in isolated products, we got molecules much larger than oxalate. And when we used carbon-13 labeled CO2, we were able to show that the products had carbon-13 in them, so we knew they came from the CO2. So we know we can get it to work with platinum. This kind of is, can we get it to work better with something cheaper kind of
Is there some hope it might get us closer to the ultimate goal that you started out with, the photosynthesis? Isn't that the goal?
Hopefully. I mean, like Dr. Roblick's used the metaphor of making a brick and we're putting it on the pile so that somebody else who continues his research can find that brick and put it in-house. So this is one step towards the ultimate goal. And it was successful, so like I said, there's a million other things you could do with this research.
I guess one thing that confuses me is I feel like they don't appear to me to be all that many more steps. It feels like you've created everything you needed to, and you just haven't quite measured if it made oxalate or something like that. I mean, why are there so many more steps? Why aren't you, just a month away from getting there?
Well, I just, mostly I wasn't able to run as many tests as I wanted to consistently. Like if I had more time, I could find ways to reproduce those compounds that weren't easy to reproduce. I could nail those syntheses down and then do tests on them. Or I could find ways to, like this, there's more than one product in that. I could find ways to really purify those and test the products separately. And with this, there might be two products in there, or they might just be the same product that's rapidly changing structure. There's a lot more to look at.
So I want to make you speculate. So let's say you had 5 years of your life to be doing this as opposed to 8 weeks, right? Where do you think the biggest barrier to actually mimicking this photosynthetic process would be through all of it?
Well, getting all of the crystal structures would be a good place to start, especially if you figuring out what each of the purifying the compounds, figuring out how to make crystals of them, and especially figuring out how to make crystals of them with CO2 bound to them. If you could figure that out, that would be a good step in the right direction. Because what if we had a crystal that had carbon dioxide bound to it, that would really help to understand what's going on. But it's not, growing crystals is such a pain. You have to test so many different things to figure out what they're soluble in, and all these different combinations of things. So that'd be where I would start, just trying to get crystals of everything.